Peace be upon you all and welcome to the channel. Continuing from the previous video, we will venture forth onto creating the plasmid map itself. So what is involved in creating a plasmid map, or actually what I should say is what's required of students when they create a plasmid map, is basically what you see in front of you here. Creating a huge circle, which is the plasmid, since plasmids normally exist as circular DNA and they are extra chromosomal, so they're not part of the actual DNA or the quote-unquote nucleus of a bacterial cell. And after drawing the circular DNA, you simply highlight the locations at which the enzymes digest the DNA strand, while pinpointing out the difference in base pairs between each cutout point. Before we start on how we actually determine where each enzyme cuts, restriction endonucleases are named after the bacteria from which they are derived from. I believe EcoR1 is obtained from E. coli. Not really sure where BAMH1 and HINE3 come from, sorry, since they're plural. English isn't my first language. I apologize for the grammatical mistakes. Yeah, so that's basically what a plasmid map involves, and we'll discuss what this is. This is basically the total number of base pairs in a plasmid and how we are able to identify that. All of that will come later on in the series. I just wanted to show everyone what a plasmid map looks like. So this should be what a plasmid map looks like. Of course, if you have the sequence of the plasmid, you'll be able to label the genes in these districts. So for example, if you have an ampicillin resistance gene or a CMV promoter or a poly A tail, that's assuming you know the sequence of the plasmid. But if you don't, you'll simply just point where each enzyme cuts, and that's about it. Now, for you to be able to determine the landmarks or the cutting points for each enzyme, you'll need a gel. So we'll switch on to this layer right here. I couldn't procure an actual gel. This is an example of an assignment that we used to take at our university. But you'll undertake the exact same steps whether you're using a paper example like this one or an actual gel. The steps are exactly the same. The only difference is that distances in this example might be a bit exaggerated compared to an actual gel. Before we start with the contents of each layer, we'll just talk quickly about what gel electrophoresis is, just so that people have a clear picture of what's going on. So, gel electrophoresis. The reason it's called a gel is because you put your DNA samples in a gel, which is created from a substance known as agarose. And the word electrophoresis, phoresis, means separation. And electro means electricity. So if you wish to define the word, and using the rule of terminology, you read the end and go all the way back to the beginning. So it's separation by electricity. And what happens during separation by electricity is that you create a potential difference across the gel. Here, this will be the negative end, and this will be the positive end. So you attach two electrodes on both sides of the gel. You run the current through, and you add, of course, you add the DNA samples here. before you run the current, and then leave it to run so that you get these DNA bands. Now, since DNA is negatively charged because of the phosphate group, it will move away from the negative end and move all the way to the positive end, because like charges repel and unlike charges attract. Now, as you can see here, each band travels a certain distance. The reason is because the DNA bands are not separated just by charge, they're also separated by size. Now, normally bigger bands tend to stay behind at the top because they cannot move as fast as the smaller bands. So think of it as a race between an elephant and a mouse. The elephant-sized bands or the large bands will stay behind because they're too slow, and the smaller mouse-sized bands move faster. It also depends on the percentage of the agarose you use in making the gel. So when you form the gel, there are pores within this gel that form when it solidifies. These pores are what the DNA uses to travel through. Now, if you wish to focus on the larger bands and disregard the smaller bands, you will need to make the pores wider so that they fit the bigger bands. And if you wish to focus on the smaller bands to separate the smaller bands and get a clearer picture, you use small sized pores. This depends on the percentage of the gel. If you use more agarose, you get smaller pores. If you use less agarose, you get bigger pores. So let's say, for example, this could be a 1% agarose gel. This could be a 0.5%. For example, I'm not saying this is what a 0.5% agarose pore looks like. I think this was, I don't remember what was the agarose percentage used for this, but it's irrelevant. So now we know what a gel electrophoresis is. Now let's start actually let's move back to plasmid mapping. So whenever you have a gel like this, first thing you need to do is label the lanes so you know what sample is in which lane. So let's start doing that. I'll zoom in. So beginning is the ladder. Hopefully I can make my handwriting small. Okay, I'll probably move a bit to the left. So this is the ladder. It's called a ladder because it looks like a ladder. It's the fiducial point for comparing band sizes. The reason we use this is because each band size is known. So this is 5,000, for example, for 4,500 base pairs, 4,000, 3,500, and so on and so forth. We will label them in a bit. So these bands are basically the ruler for the bands in the unknown lanes. So for example, if you want to know how big this band is, you simply compare it to your ruler. So if this is 4.5, this is 3,500 base pairs. So since this is at the same level, this automatically becomes 3,500 base pairs. The problem lies within the base pairs that lie between the two increments like this. So this is 1,000, this is 500. We don't know what this is. So the ladder's purpose here is to create what we call a standard curve so that we can extrapolate the band size here. And we'll discuss standard curves in the next video. we'll just take the steps that will lead us to the standard curve. This lane contains an uncut plasmid. Honestly, I do not know why it's included in the assignment because you really do not benefit from it at all, but I guess it's to show supercoiling, because the only reason you'll get two bands is if this is the heavier band, then the DNA supercoils, becomes smaller, and then you get another band emerging at the bottom. But other than that, you do not use it whatsoever with regards to creating a plasmid map. So I'll just okay, I probably won't interfere with the labeling here, so we can continue. Then these three lanes, 1, 2, and 3, contain single restriction endonucleases. So we put the plasmid combined with one enzyme, then in the plasmid with the second enzyme, and the plasmid with the third enzyme. So since there's only one enzyme in each sample, this is what we call a single digest. Now let's see this enzyme. The enzyme here with the sample was ECO. I won't add the R1 to give my writing some space. I have big handwriting, unfortunately. And this, if I remember correctly, was BAM, and this was HIND. There is a legend at the bottom. I'll just use my memory for now. This is ECO and BAM, if I recall. This is ECO and HIND. And the last one is BAM and HIND. Oh yes, these three remaining lanes have two enzymes with the plasmid instead of one. So these are the single digest lanes, these are the double digest lanes. So you have plasmid plus ECO plus BAM. Here plasmid plus ECO plus HIND. And here is plasmid plus BAM plus HIND. Let me just check the order. So ECO, BAM, HIND, ECO, BAM, ECO, HIND, BAM, HIND. Yes, we're good to go. Just zoom out just a little bit. So now we know what was placed in each lane. So let's take a look. Here ECO gave us two bands. That means that ECO, or ECHO depending on how you would like to pronounce it, cut the plasmid twice to give you two bands. BAM gave us three bands, which means three cuts. HIND gave us only one band, which means that this band is the full size of the plasmid. We'll draw these two and how ECO and BAM cut to give you here two bands and three bands respectively. But HIND here cut the plasmid once and gave us one band. And this band right here, this is the full size of the plasmid. Now how do we know that? I'll show you a small diagram here. So this is the plasmid, the circle, and HIND cuts here, let's say. So HIND. When you cut circular DNA, it automatically forms a linear DNA. Now this linearity could be observed on the gel as this one. So since the entire plasmid is preserved or conserved after the cutting here, this band, or rather this band, tells us that the full size of the plasmid is, well, it matches here, so that means the full size of the plasmid is around 5,000 base pairs. But we'll do the standard curve just to confirm. Now BAM cuts twice, 3 times, sorry, and ECO cuts twice. We'll take a look at all 6 diagrams of these lanes in the next video. For now, we'll just move on to the next step to create the standard curve. So I'll just go ahead and label the ladder with the base pairs. So this is 5,000 base pairs. I'll write the unit here. I always confuse it with boiling point. This is 4,500 or 4,500. It always decreases by a 500 increment. This is 4,000, 3,500. Probably zoom in so you can see better. This is 3,000. This is 2,500. This is 2,000. Let's drag this up. This is 1,500. 1,000. And then we come to the smallest one, 500. So that's a standard. The ladder is called the standard because we already know the sizes of each bands, and that will help us with the creation of the standard curve. Now, how do we create the standard curve with this knowledge? What you need to do is measure the distance traveled of each band, then recording it. And I'll show you what to do with this information after that. So the way you would measure the distance traveled, if you're using a software like Photoshop or Clip Studio, like the one I'm using currently, which was known in the past as Manga Studio, I believe there's some sort of a digital ruler where you can place a point and then drag it to create a line, and then it will tell you how many pixels or how many centimeters it is. Unfortunately, I am new to digital art, so I normally print this paper out and measure it with a ruler. You can also do the exact same thing with an actual gel. It will probably be a bit small depending on the size of the gel you use, if you're using a big cast or a small cast or a mini gel. So what you do is you measure starting with the bottom edge of the lane right here. So you start from here. This is where you place the 0, and then you measure all the way down to the bottom edge of each band like this. So you'll measure from here all the way to here and then record the distance. Sorry, that was supposed to be a straight line. And record from here, for example, all the way to the bottom edge. From the bottom edge of the lane where you place the samples all the way to the bottom edge of each band, and record these distances. I have done this beforehand for this demonstration, so I'll show you the distances for the ladder. They're right here. Oh, actually I recorded the distance for all the lanes, which is good. So as you can see here, I measured the bands, I measured the ladder, then I measured we're ignoring the uncut plasmid, so we will not resort to that in any form. Then I recorded the eco lane, the band, the hind, and then the double digest lanes. Oops. So now we're ready to start with the standard curve. So I will end the video here, and the next video we will start with our Excel sheet and how to plan our standard curve and where to go from there onwards. Thank you very much for your time. Be safe and take care.